Did you know that scientists are now making something called golden rice to fight vitamin A deficiency?
Yeah, golden rice is pretty incredible.
And they're using like spider silk genes in goats to create these super strong materials.
It's pretty wild what they're doing with biotechnology these days. But it's also important to remember that it all builds on a long history of humans working with nature.
Oh yeah, totally.
Like, think about things like making curd, bread, wine. These are like early forms of biotechnology, even though they're way simpler than the stuff we're seeing today.
That's a great point. It's like we've been doing biotech for centuries, but now we're taking it to a whole new level. It seems like genetic engineering and bioprocess engineering were like the two techniques that really launched modern biotechnology.
Yeah, for sure. Those two really changed the game. And genetic engineering, especially, manipulating the DNA and RNA, the stuff that makes life tick, that's what lets us create organisms with specific traits. It's like having a toolkit at the molecular level where you can actually edit the blueprints of living things.
So instead of traditional hybridization where you're kind of just, mixing things up and hoping for the best, genetic engineering is like super precise.
Exactly. You can pinpoint a gene that gives, say, pest resistance and put it directly into a plant's DNA.
Okay, that's mind-boggling. The YouTube lecture talks about using something called a plasmid to do this. Honestly, it sounds a bit sci-fi to me.
Yeah, plasmid does sound kind of futuristic. Think of it as a delivery vehicle for genes. It's this little circular piece of DNA that can carry foreign DNA into a host cell.
So it's like reprogramming an organism, giving it new ability.
Exactly. The applications are huge.
The lecture throws around a bunch of technical terms,
Mhm.
restriction enzymes, polymerase enzymes, ligases, vectors, host organisms. I'm not going to lie, it's a lot to digest.
Yeah, I get it. It can seem pretty overwhelming at first. But really, these are just the tools that make genetic engineering possible. Each one has its own job in the whole process of manipulating and transferring DNA.
Okay, so it's like an assembly line, but for genes.
Yeah, kinda. Like, for example, restriction enzymes, these act like molecular scissors, cutting DNA at very specific sequences. And these cuts create something called sticky ends, which is where you can stick in new DNA fragments.
Sticky ends. Okay, that sounds a little messy.
It's actually super precise. These sticky ends let the DNA fragments connect really easily, kind of like puzzle pieces fitting together. And believe it or not, this precision comes from palindromes in the DNA sequence.
Wait, palindromes like race car or madam, what do those have to do with DNA?
Same concept, but with DNA. A palindromic sequence in DNA reads the same forwards and backwards. Restriction enzymes, they spot these palindromes and cut the DNA right within them, creating those sticky ends so everything joins perfectly.
Oh, wow, that's really cool. So it's all about precision cutting and pasting at the molecular level.
Exactly. And to see these teeny tiny DNA fragments, scientists use something called gel electrophoresis. It separates them by size, so you can pick out and isolate the specific ones you want.
Gotcha. So once you have that DNA fragment, how do you actually get it into a host organism?
Ah, that's where cloning vectors come in. They're like vehicles carrying that foreign DNA into the host cell. Plasmids, which we talked about earlier, those are a common type of cloning vector. And the cool thing is, they can replicate independently inside a host cell, making tons of copies of the DNA you put in there.
So it's like hijacking a cell system to make more of the DNA you want. But how do they make sure that only the cells with that new DNA survive?
They use things called selectable markers, which are often genes that make cells resistant to antibiotics. It's kind of like trying to find a needle in a haystack. If you could make all the hay disappear except for the needle, it would be much easier, right? That's what these markers do.
That's a great analogy. It's like giving the transformed cells this superpower to survive in a tough environment.
Exactly.
Now, both our sources talk a lot about biotechnology and agriculture, especially with the global population growing and, resources getting scarcer.
Yeah, definitely. We need to get creative to feed everyone sustainably. The sources talk about three main approaches: agrochemicals, organic farming, and genetically modified crops, GMOs.
Sounds like a three-way race to boost food production. Let's break them down one by one, starting with agrochemicals.
Okay, so agrochemicals like fertilizers and pesticides, they can definitely give crop yields a quick boost, but they can have some long-term downsides for the environment.
Right.
Overuse can lead to polluted water, harm beneficial insects, and even make pests resistant to the pesticides. And they can be pretty expensive for farmers, too.
Yeah, it seems like a bit of a band-aid solution, maybe with some unintended consequences down the line. What about organic farming? That feels like the more eco-friendly option, right?
It is, but it can also be slower and less efficient than conventional methods, so it can be hard to produce enough food for everyone just relying on organic farming.
So each approach has its own set of pros and cons. It seems like our sources are pretty excited about GMOs, though. What makes them so promising?
Well, with GMOs, scientists can introduce specific traits into crops much more precisely and quickly than with traditional breeding. Like, for example, they can make crops resistant to pests, tolerant to drought, or even packed with more nutrients.
I remember learning about the Green Revolution, which was this huge increase in food production in the mid-20th century.
Hmm.
But didn't that rely heavily on those agrochemicals we were just talking about, which we now know can have some negative effects?
Exactly. That's why GMOs are seen as a potential solution to some of the problems created by the Green Revolution.
Right.
They can increase yields, but also reduce our dependence on harmful chemicals, which is a much more sustainable approach.
Okay, I'm starting to see why GMOs are generating so much buzz. Can you give me some concrete examples of how they're being used to actually benefit agriculture?
Of course. One really interesting example is golden rice. It was developed to combat vitamin A deficiency, which is a huge issue in many parts of the world where rice is a staple food.
Golden rice. It sounds almost mythical.
It is a pretty simple concept, but with huge potential to change lives. Scientists took a gene from daffodils that produces beta carotene, a precursor to vitamin A, and they put it into rice plants. And what you get is rice with a golden hue and, most importantly, much higher levels of vitamin A.
So eating this rice can actually help prevent vitamin A deficiency and all the health problems that come with it. That's amazing.
Exactly. And it shows how biotechnology can tackle those nutritional deficiencies, especially in places where people don't have access to a very diverse diet.
It's a really cool example of the positive impact GMOs can have. Did you find any other examples in the sources that show how biotechnology is tackling agricultural issues?
Definitely. Another success story is Brait cotton. This genetically modified cotton is resistant to certain insect pests that belong to the order Lepidoptera.
Lepidoptera, those are butterflies and moths, right? Have you ever thought of them as being a threat to crops?
Well, it's their larvae, the caterpillars, that are the real problem. They can cause a ton of damage. But cotton was created to fight this problem using a gene from a soil bacterium called Bacillus thuringiensis, or beet for short.
Wait, they're fighting pests with bacteria. How does that even work?
Beet naturally produces a protein that's toxic to certain insects, but it's totally harmless to humans and other animals.
Okay.
So scientists took the gene for this protein called the cry gene and put it into cotton plants.
Mm.
And when the caterpillars munch on these plants, they get a dose of this toxic protein, which messes up their digestion and, well, they don't make it.
Wow, so it's like targeted pest control, but at a genetic level. That's way more sustainable than just spraying insecticides everywhere.
It is. And what's even cooler is that the cry genes used in Brett Kari are specific to certain types of insects. Scientists can pick a cry gene that targets the exact pests that are bothering the crops so they're not harming beneficial insects or anything like that.
That's super clever.
Yeah, it really highlights how precise genetic engineering can be.
Okay, beet cotton. That's another great example of the potential of GMOs. Did the sources mention any other ways biotechnology is being used to solve those agricultural puzzles?
They talked about this really cool technique called RNA interference, or RNAi.
RNAi, okay.
It's this natural defense mechanism that plants and animals use to control things at the cellular level, and scientists have figured out how to harness it to control pests.
Wow.
One example they gave was using RNAi to combat nematodes, these tiny worms that attack the roots of tobacco plants.
So these microscopic worms are causing big problems for tobacco.
They sure are. They cause something called root knot disease.
Yep.
And the way RNAi comes into play is by silencing certain genes. So nematodes, they make certain proteins that damage the tobacco roots. Scientists can use RNAi to basically shut down the genes that make those harmful proteins.
So it's like cutting off the supply line, preventing the nematodes from producing their weapons.
Exactly. And the way they do this is by putting genes into the tobacco plants that produce double-stranded RNA molecules. And these molecules trigger the RNAi mechanism, which goes around and destroys any RNA that matches that double-stranded RNA.
So it's like a seek and destroy mission at the molecular level.
Yeah, pretty much.
Yeah.
By adding these special genes, they're giving the tobacco plants a way to fight back against the nematodes.
And they're using Agrobacterium tumefaciens again to do this, right? The bacteria that naturally transfers DNA to plants.
Yep, same one. It's another example of scientists taking advantage of natural processes to solve agricultural challenges.
It's really amazing to see how biotechnology is being used to make agriculture more efficient and, better for the planet.
Yeah.
Golden rice, food cotton, RNAi. It's incredible what scientists are doing.
Yeah, absolutely. And that's just a small sample of what's out there. The possibilities are really endless, and scientists are constantly coming up with new and innovative ways to tackle food security issues.
These examples have been super insightful. It's clear that biotechnology has a ton of potential in agriculture. But let's switch gears a bit now and dive into its applications in medicine, which our sources also cover in a lot of detail. It seems like biotechnology is revolutionizing healthcare, particularly in the development of new therapies.
It is. One of the most remarkable things biotechnology has done for medicine is the production of recombinant therapeutics. These are basically proteins that are made using genetically modified organisms, and they've changed the way we treat so many diseases.
So they're using living organisms as factories to produce medicines. That's wild. What are the advantages of using this approach?
Well, for starters, recombinant therapeutics can be produced in huge quantities, which makes them more accessible and affordable. They're also very specific and effective, targeting the actual cause of a disease with a lot of precision.
And because they're often derived from human genes, there's less chance of the body rejecting them.
Right, exactly. It's like giving your body a boost with its own natural defenses, but in a more targeted and powerful way.
I'm starting to understand why these recombinant therapeutics are such a game changer. Can you give me some examples of how they're being used to actually treat diseases?
Sure. One of the most well-known examples is humulin, which is recombinant human insulin. Before genetic engineering, people with diabetes had to use insulin extracted from animals, which often caused allergic reactions.
So humulin was a huge breakthrough for people with diabetes. How did they even make human insulin in a lab?
They used bacteria. Scientists isolated the gene for human insulin and put it into E. coli bacteria. And these bacteria basically became little factories churning out human
So
insulin.
these microscopic pharmaceutical companies?
Pretty much. But, insulin's a pretty complex protein. It's not as simple as just putting the gene in there.
Right.
Insulin has these two chains of amino acids that have to be linked together in just the right way. Bacteria don't naturally make those chains in the correct form, so scientists had to get a bit creative. They engineered the bacteria to produce each chain separately and then combine them in the lab to make functional insulin.
That's some serious molecular wizardry. It's incredible how they can work around nature's limitations to create these life-saving medicines.
Yeah, it is pretty amazing. And humulin is just one example. There are now recombinant therapeutics for things like hepatitis B, growth hormone deficiencies, and even some types of cancer.
It seems like there's no limit to what they can do. It sounds like recombinant therapeutics are really changing the face of how we treat diseases.
They absolutely are. And we're really just at the beginning. As our understanding of genetics and biotechnology keeps growing, we can expect to see even more incredible therapies in the future.
That's exciting to hear. Our sources also mention gene therapy as a promising application of biotechnology in medicine. Can you explain what that is and how it works?
Sure. Gene therapy is basically replacing a faulty gene with a healthy one to treat a genetic disease. It's like fixing a broken blueprint in the body to restore its ability to function properly.
That sounds almost like science fiction. Is it really possible to edit genes inside the human body?
It is. One of the earliest success stories involved a young girl who had severe combined immunodeficiency, or SCID. This is a condition caused by a defect in a gene called adenosine deaminase, or ADA.
Okay.
And this gene is super important for developing a healthy immune system.
So her immune system was basically compromised from birth.
Yeah, it was.
How did gene therapy help her?
Well, before gene therapy, the only treatments for SCID were frequent enzyme injections or a bone marrow transplant, and both of those came with some serious risks. Gene therapy offered a potential cure by actually replacing that faulty ADA gene with a healthy one.
So they reprogrammed her cells to produce the missing enzyme. How did they pull that off?
They took lymphocytes, which are a type of white blood cell, from her bone marrow. Then, using a retrovirus as a vector, they delivered the correct ADA gene into those cells.
Hold on, a retrovirus. Aren't those the kinds of viruses that cause diseases like HIV?
They can be, but scientists have figured out how to modify them and make them safe to use. In this case, the retrovirus was like a delivery truck carrying the good ADA gene into the lymphocytes.
It's like turning an enemy into an ally. That's pretty amazing.
Yeah, it is. So, after they delivered the gene, they multiplied these modified lymphocytes in the lab and then injected them back into her bone marrow.
Okay.
And these modified cells started producing the missing ADA enzyme, which essentially restored her immune system's function.
Wow. So they gave her body the tools to heal itself.
Wow.
Was this a permanent cure for SCID?
In her case, it wasn't a permanent fix because the modified lymphocytes eventually died off.
Oh.
But if gene therapy is done during the embryonic stage, when cells are still dividing rapidly, then it can actually lead to a permanent cure.
So it's all about timing.
Yeah, timing is crucial.
Gene therapy sounds like a revolutionary way to treat genetic diseases.
It really is. It's still a pretty new field, and there are definitely challenges to overcome. But the potential benefits are huge, and scientists are constantly pushing the boundaries of what's possible.
That's incredible.
Mhm.
Humulin and gene therapy are such amazing examples of how biotechnology is transforming medicine. Our sources also briefly touch on molecular diagnosis. Can you explain what that is and why it's so important?
Molecular diagnosis is all about detecting the root cause of a disease at the molecular level. It's like zooming in on the specific genes or molecules that are malfunctioning, like using a super high-tech magnifying glass.
It sounds like high-tech detective work. How is it different from those traditional diagnostic methods?
Well, traditional methods often rely on detecting the symptoms of a disease, which might not show up until it's already pretty advanced.
Right.
But molecular diagnosis lets us see those underlying genetic or molecular abnormalities that are causing the disease, sometimes even before any symptoms appear.
So it's like catching a criminal before they even commit a crime. Our sources talk about some specific techniques used in molecular diagnosis. Can you break those down for us?
One that you've probably heard of is PCR, or polymerase chain reaction.
Oh, yeah, PCR.
It's a way to amplify tiny amounts of DNA, making it possible to detect even the tiniest traces of a pathogen or a genetic mutation.
We talked about PCR earlier when we were discussing genetic engineering. I didn't realize it had so many uses in diagnostics, too.
Yeah, it's super versatile. It's used to detect things like viral infections like COVID-19,
Right.
and it's even used in fields like paleontology to analyze DNA from ancient fossils.
Wow, PCR is helping us understand both the present and the past. That's pretty mind-blowing. What other molecular diagnostic techniques are there?
Another one is ELISA, which stands for Enzyme-Linked Immunosorbent Assay.
Okay.
It's a really powerful tool for detecting specific proteins, including antibodies.
Right. Antibodies, those are the proteins our immune system makes to fight off invaders.
Exactly. And ELISA takes advantage of the way antibodies bind to their specific targets called antigens.
Okay.
So, for example, in an ELISA test for HIV, a plate is coated with HIV antigens. And if a person's blood sample has HIV antibodies, those antibodies will bind to the antigens on the plate.
It's like a trap for the HIV antibodies.
Yeah, you could say that.
But how do you actually see if this binding is happening?
That's where the enzyme-linked part comes in. The test uses another antibody that's specific to human HIV antibodies, and this antibody has an enzyme attached to it. So when this enzyme-linked antibody binds to the HIV antibodies, it sets off a color change that's easy to see.
So it's like a chain reaction at the molecular level, but it results in a signal you can actually see.
Exactly.
That's super clever. Are there any other molecular diagnostic tools in the biotech toolbox?
Our sources mention autoradiography, which uses radioactive probes to detect specific DNA sequences. This is really helpful for identifying genetic mutations.
Radioactive probes, that sounds kind of intense.
Well, think of it like this. Imagine you have a blueprint with a tiny error that's causing a problem.
Okay.
A radioactive probe is like a highlighter that attaches to that specific error, making it easy to spot.
Ah, so it's like a molecular spotlight. How does it actually work?
Scientists create a radioactive DNA or RNA probe that matches the normal gene sequence they're interested in. And then they let this probe bind to a sample of the patient's DNA.
Okay, so if the patient's gene is normal, the probe will bind perfectly.
Exactly. And then when you expose it to a special kind of film, the radioactive probe will create a signal showing you exactly where it is.
Okay.
But if the patient has a mutation in that gene, the probe won't bind properly and there won't be a signal.
It's like a molecular puzzle. If the pieces fit, you get a signal. If not, something's off.
Yeah, that's a great way to put it.
It's pretty amazing.
And by analyzing these patterns, scientists can figure out exactly where a genetic mutation is and what kind it is, which helps them diagnose and understand all kinds of diseases.
PCR, ELISA, autoradiography. These molecular diagnostic techniques are really remarkable. They're giving us a window into the inner workings of our bodies at such a detailed level.
I know, right? And the cool thing is, these techniques are constantly evolving. As we learn more about genetics and molecular biology, we can expect even more sophisticated and powerful diagnostic tools in the future.
This has been such an informative journey into the world of biotechnology. We've learned so much about how it's applied in agriculture and medicine, but we've only scratched the surface. Up next, we'll be exploring the fascinating realm of transgenic animals.
Welcome back, everyone. Get ready to dive into the world of transgenic animals. These are animals whose genes have been tweaked, either by adding new genes or deleting existing ones.
Transgenic animals. That sounds straight out of a sci-fi movie. I mean, why go through all that trouble to create them?
While transgenic animals have become incredibly useful in research, they can be used for all sorts of things, like studying human diseases, testing new drugs and vaccines, even producing therapeutic proteins in their milk.
Wait, hold on. Therapeutic proteins in milk. What, like protein shakes with superpowers?
Not exactly. Think more like specialized medicines. Our sources mention a transgenic cow named Rosie that was engineered to produce milk containing human alpha-lactoalbumin.
Oh, okay.
That's a protein that's really beneficial for newborns.
So they basically turned Rosie into a living medicine factory, making baby formula with extra benefits.
That's a great way to put it. It's a perfect example of how transgenic animals can be used to produce important proteins for medical uses. And Rosie's not the only one. Scientists have also created transgenic sheep that produce alpha-1 antitrypsin in their milk.
And what's that used for?
It's used to treat emphysema.
Right, emphysema. That's a serious lung disease.
Yeah.
How does that protein help with that?
Well, emphysema is often caused by a deficiency in alpha-1 antitrypsin, which is a protein that protects the lungs from damage.
Okay.
So transgenic sheep, like the one our sources mention, they can provide a whole new source of this essential protein.
That's pretty amazing.
It is.
So are mice the only animals being used for this kind of stuff?
Mice are definitely the most common because they're small, easy to breed, and we understand their genetics really well.
Right.
But scientists are using this technology with other animals too, like sheep, rats, rabbits, pigs, goats, even monkeys.
Wow, that's a pretty diverse group. It seems like the possibilities are endless. But I guess stepping back a bit, why are transgenic animals so valuable for research in the first place?
They give us a unique way to study human biology and disease.
Okay.
Because we share a lot of genes with other mammals, studying transgenic animals can help us understand how those genes work and what happens when they don't work properly.
So it's like having a living, breathing model of a human disease.
Exactly. Scientists have created transgenic mice that develop things like cancer, Alzheimer's disease, even obesity. By studying these animals, they can get insights into how these diseases progress and hopefully find new treatments and cures.
So transgenic animals are helping us understand and treat human diseases. But what about applications outside of medicine?
Oh, there are lots of potential applications in agriculture too. For example, scientists are looking into using genetic modification to enhance livestock breeding.
Oh.
So they can create animals that are more resistant to diseases, produce more milk or meat, or even have better nutritional content.
It's like selective breeding, but on fast forward.
Yeah,
There are
exactly.
more waiting generations for those desirable traits to show up.
Exactly. Transgenic technology brings a lot more precision and speeds up breeding programs significantly.
Wow.
And it's not just about efficiency, it can also have a big impact on animal welfare.
Okay.
Imagine chickens that are resistant to, like, avian influenza. That would be a win-win, both for the farmers and for the chickens themselves.
That's a great point. It's not just about making things more efficient, it's about making things better for everyone involved.
Mm.
So, transgenic animals are making a splash in medicine and agriculture. What about other industries?
Believe it or not, transgenic animals are even being used in industrial applications. One of our sources talks about a company called Nexia Biotechnologies that made transgenic goats that produce spider silk proteins in their milk.
Hold on, spider silk and goat's milk. That's a combination I never would have thought of. What's so special about spider silk anyway?
Well, spider silk is incredibly strong and flexible.
Right.
It's actually stronger than steel, ounce for ounce.
Wow, I had no idea.
Nexia Biotechnologies took the genes that make spider silk and transferred them into goats. So now they have goats producing milk that's full of those valuable spider silk proteins.
That's pretty mind-blowing.
It is.
So what are they doing with all that spider silk protein?
They're using it to make all kinds of materials with amazing properties.
Okay.
They've developed fibers that are stronger and lighter than conventional materials. And these fibers could be used for just about anything, from tennis rackets and bulletproof vests to surgical sutures and artificial ligaments.
It's like they're blending biology and engineering. So we've seen how transgenic animals can be used in medicine, agriculture, even industry. But with all this genetic manipulation going on, shouldn't we be thinking about the ethical implications?
Absolutely. Our sources touch on bioethics, biopatents, and biopiracy, all of which raise important ethical questions in biotechnology. Let's start with bioethics.
Okay, what exactly is bioethics?
Bioethics is basically a set of moral principles that guide research and applications in biotechnology. It's about making sure these powerful tools are used responsibly and ethically.
Right.
We need to think about the potential impacts on human health, the environment, and society as a whole.
So it's like a moral compass for scientists, helping them navigate all the tricky ethical issues that come up in this rapidly changing field.
Exactly. Biotechnology has the potential to do a lot of good, but it also raises some big ethical questions like, is it okay to modify the genes of animals for our own benefit? What are the risks of releasing genetically modified organisms into the environment? Bioethics gives us a framework for thinking through these tough questions.
The sources mentioned a specific organization in India called GEAC.
Yes.
What is that and what role does it play in all of this?
GEAC stands for Genetic Engineering Approval Committee.
Oh, yeah.
It's a government body in India that's in charge of evaluating and approving any research proposals that involve genetic engineering. They assess the safety and ethical implications of the research before giving it the green light.
So it's like a safety net, making sure that any research involving biotechnology is done responsibly and doesn't pose unnecessary risks.
Exactly. GEAC plays a crucial role in regulating biotechnology in India.
That makes sense. So bioethics and regulatory bodies like GEAC are essential for making sure that biotechnology is used responsibly. But what about biopatents? How do those fit into all of this?
Biopatents are legal rights that are granted to inventors for their discoveries or inventions in the field of biotechnology.
Okay.
They give inventors control over how their inventions are used and commercialized, which can be a big incentive for innovation.
So it's like a patent for a new gadget, but for a biological invention.
Precisely. Biopatents play a really important role in the biotechnology industry, encouraging investment in new technologies. But they can also be controversial, especially when it comes to patenting genes or other natural biological materials.
Yeah, that does seem like a tricky area. So biopatents are about protecting intellectual property and encouraging innovation. What about biopiracy? That sounds a lot more sinister.
Biopiracy is the unauthorized and unethical exploitation of biological resources. It often happens when multinational corporations from developed countries take advantage of countries that are rich in biodiversity but don't have the resources to protect their biological heritage.
So it's like companies swooping in and stealing natural resources from developing countries without permission or fair compensation.
Unfortunately, that's often the case. Our sources mention a few examples, like attempts to patent basmati rice, turmeric's wound healing properties, and neem's antimicrobial properties. All of these are things that have been known and used in India for centuries, part of their traditional knowledge systems.
It sounds like they're not just stealing resources, they're stealing knowledge and claiming it as their own. Did anything good come out of those situations?
Luckily, India was able to challenge those cases of biopiracy and get back its rights over those resources and its traditional knowledge.
That's good to hear. It seems like there's this constant battle to protect biological heritage from exploitation.
There is, but it's a fight worth fighting. These resources and knowledge systems are incredibly valuable and deserve to be protected.
We've covered so much ground today, from the basics of genetic engineering to the ethical dilemmas of biotechnology. It's such a fascinating field, but it's also really complex. Where do we even go from here? I feel like we've just scratched the surface of this whole biotechnology thing.
I know, right? It's just amazing how fast this field is moving and all the incredible things that are being done. It feels like we've covered so much in this deep dive, from like the nitty-gritty of genetic engineering to the crazy applications in agriculture and medicine, and then, all the ethical stuff that comes along with it.
Totally. It's been such a trip exploring how biotechnology is like shaping our world. We've talked about everything from tiny genes to like those transgenic animals making medicines in their milk,
Yeah.
and even things like biopiracy. It's clear that biotech has the power to do a lot of good, but it's also clear that we have to be really careful with how we use it.
Yeah, absolutely. Biotechnology isn't just about science, it's tied to our values and like what kind of future we want to build. As we keep learning more about life and how to manipulate it, we have to be humble and aware of the potential consequences. We can't just charge ahead without thinking about the ethical side of things.
This deep dive has definitely opened my eyes. I'm still amazed by how much potential biotechnology has to solve some of the world's biggest problems. But I'm also more aware than ever that we need to keep talking about the ethical stuff and make sure that these powerful tools are being used to benefit everyone, not just a select few.
I couldn't agree more. Knowledge is power, but it's what we do with that knowledge that really matters. We need to keep having these conversations, think things through carefully.